Good afternoon, everyone. Thanks for coming to my thesis defense. My name is Marion Tredez, and I'm going to present you a transcriptomic analysis of the Mediterranean blue mussel during its ontogeny. This work was carried out within the EVO Inside team of the Developmental Biology Laboratory of Differential under the supervision of Michael Schubert. You probably know them. Steamed, creamy, with curry, mussels are not only flagship species for gastronomy, they are also valuable indicators of the ecosystem health. As sentinel species, they accumulate the pollutants by filtering the water to feed on plankton and organic waste, giving thus information about the water quality. As part of the bivalves class, with their shell composed of two calcareous valves dorsally hinged, they can also be impacted by ocean acidification that make them potential indicators of the climate change. Ideal for aquaculture, with their wide distribution and flexibility, Mytilus galloprovincialis is also invasive worldwide. Attaching to rocky substrate, it has a high resistance to desiccation, a high fertility, and important growth and recruitment rates. In addition of this economical and ecological interest, these characteristics make this species a promising laboratory model. This non-model species has already been the subject of some studies, but most of them ecotoxicological and focusing exclusively on the adult stage. Nothing is known at molecular level on its ontogeny, which means its development. What is known about this species is its life cycle, common to all the mussels, with a pelagic phase starting with the fertilization of the gametes, developing into multiple larval stages, and a benthic phase starting with the loss of the swimming ability and the settlement of the mussel. With the boom of the omics technologies these last 20 years, numerous opportunities have arisen to make progress, particularly in the fields of evolution and development. The genome of the Mediterranean mussel has been sequenced, and in this recent paper of 2020, they found a high individual variability, with about 1/3 of the genes subject to presence-absence upon individuals. The objective of my work was to start filling the molecular gaps on this species ontogeny by using a transcriptomic approach. Indeed, focusing on mRNAs instead of DNA or proteins can highlight gene expression differences between samples and also can highlight genetical differences between individuals. But what to do after the RNA sequencing? Two bioinformatical methods exist. The genome-guided assembly. So basically, it's a method that is aligning the RNA sequenced fragments on a known reference genome from the same species. And the second method is the de novo, that is aligning the RNA-seq fragments based on overlapping between them. My work, therefore, aimed to assess. So, for the de novo analysis, there are potential limitations. As you see, so with the de novo, it's not guided by an a priori, it's not guided by the reference genome. It can have potential limitations. It is known to be less efficient than the genome guided. And also, the individual variability that can be highlighted by the de novo cannot be highlighted with the genome guided, because in the genome guided method, this individual variability corresponding to the RNA fragments that are not aligned to the reference genome, they are removed during this analysis. My work, therefore, aimed to assess if the de novo assembly was as efficient than the genome guided, given the individual variability of this species, and also to determine if the transitions from one developmental stage to another are well reflected by the transcriptomic profiles. To answer these two questions, adult mussels were collected in the Bay of Villefranche-sur-Mer that you probably know. They were maintained in aquaria, and the spawning was induced by a heat shock. The larval pool obtained from the 4 different parental pairs were pooled 2 per 2 in order to obtain a biological duplicate, P2 and P3, with different genetical backgrounds. RNA extractions have been done on 15 different time points, from the 0 hours post-fertilization corresponding to unfertilized egg to 72 hours post-fertilization corresponding to the D-veliger larvae, characterized by its D shape. The results after this RNA extraction, so the RNAs have been sent to the sequencing, an in silico analysis has been performed, as well as an experimental validation. Let's go into details about the in silico analysis, which means basically the computer things. The first thing you have to know is that a lot of tools exist today to perform genome-guided and de novo analysis. The tools that I choose, written in green on this scheme, have been chosen according to their speed and their memory consumption, as well as their efficiency according to online tutorials and literature. So the first step, the RNA sequencing results are resumed in FASTQ files. The first step is to determine the quality of the sequencing by analyzing the FASTQC reports. Then the next step is to reassemble the different RNA fragments into transcripts. I will first explain you the genome-guided method. I first use STAR to align the reads, so the RNA-seq fragments, to the reference genome from 2020 that I told you before. Then string-tie was used to count the number of transcripts that were contained in each sample. BLAST was used to determine to which protein the transcript found were corresponding to. So I blasted it on an annotation file that is available from the reference genome paper. And then I built a matrix with the number of counts that are indicated, with the number of counts for each transcript in the lines and for each samples in columns. And a differential expression analysis was performed from this matrix. So basically, it's the building of graphs and making statistics. Concerning the de novo assembly now, I use Trinity. As I told you, the de novo assembly is known to be less efficient because not guided by an a priori. So the quality of this assembly had to be assessed using Bowtie and Busco. I will explain you what they do just after. I also had to perform filtrations. First, to remove the transcripts that didn't match with a known protein within the metazoans. And I had to apply another filter to remove redundant information within this transcript. Then I used Callisto for the same reason than string-tie to count the transcript. And for the rest, I did exactly like for the genome-guided method. Let's first have a look to the quality of the de novo assembly. The Bowtie results indicate that less than 2% of the RNA fragments didn't match with another one and were thus not part of the assembly. The Busco results indicate that 99% of the found transcripts found an equivalence in the pool of conserved proteins within the metazoans. So basically, these two numbers indicate a particularly efficient de novo assembly that was able to retrieve a huge number of transcripts even after the transdecoder filtration. You can see that you have 30 times more transcripts found in the de novo compared to the genome-guided. And after filtering sequences that are 90% and 99% redundant, respectively, you can see that you still have a higher number of transcripts than the genome-guided. This additional information could correspond to the dispensable genes identified by the reference genome paper and could potentially correspond to individual variability. That was one of the major limitations of the genome-guided methods. But now you have this in silico result. How do they fit to the reality, the experimental reality? I performed a quantitative validation by using quantitative polymerase chain reaction. So basically, this method is quantifying the expression of a gene based on its expression monitoring, on its amplification monitoring over time. I'm showing you here the result for chitin synthase. That is an enzyme involved in the shell secretion in bivalves. And as you can see, the de novo, the qPCR, and the genome-guided patterns of expression of this enzyme are coherent. You have an increase to reach a peak at 44 hours post-fertilization. This corresponds to the literature saying that the shell is developing within the first 48 hours. And it is also coherent given the fact that the organ secreting the shell is starting to develop at 24 hours post-fertilization, the point from where you see the slope that becomes steeper compared to the first phase. The chitin synthase expression can also be observed qualitatively, so the spatial distribution of the mRNAs inside the larvae. For that, we can use in situ hybridization. I used in situ hybridization chromogenic and fluorescent. So basically, in the in situ, you use probes that are complementary to your mRNA, and it allows you to visualize its expression pattern. So first of all, you can see that for the same time point, the chromogenic and the fluorescent give a coherent result. And also, you observe that the chitin synthase expression seems to start in the hinge part, so the flattened dorsally part of the larvae, that is coherent with the shell field that starts its development in, say, the dorsal hinge part. And for the D-Veliger pattern of expression, you can see that the margins are labeled. And this is coherent also with the literature, because when they inhibited this enzyme, they found a perturbation of the larval shell at these margins and at this hinge. These methods, in silico, qualitative, quantitative, they aimed at starting filling the gaps on the med muscle ontogeny. Talking about the development, I performed clustering. These clusters are representing dissimilarities and correlations between the different samples. As you can see, with the genome-guided method, the biological duplicates P2 and P3 were well clustered together for each time point. While in the genome-guided, the biological duplicates were only clustered 3 times and with a much lower p-value indicated in red, the numbers. And this clustering of the biological duplicates in the genome-guided assembly potentially allowed the distinguishment of 4 different developmental periods: the embryo, the gastrula, where the 3 embryonic layers are formed, the trochophore, where the shell starts being secreted, and the D-Veliger, where the major organs are well formed. To conclude, we could say that the de novo assembly was as efficient than the genome-guided method because 97% of the transcripts were commonly found by both techniques, but we could also say that the de novo was more efficient than the genome-guided since it was allowed to bring new information that are potentially isoforms and individual variability. So the de novo can appear more efficient, but pay attention, the de novo requires more computational power. It was really heavy and really low to process. So according to the resources you have, according to the time you have, but even more importantly, the question that you have to address, both methods could be considered as efficient. For example, to answer the second question about the different developmental stages distinction, the genome-guided was more efficient because the de novo, with the individual variability retrieved, it could have distributed the duplicate clustering and the developmental periods distinction. This preliminary study, also requiring further investigations, provided successful in silico and experimental transcriptomic methods. It also provided valuable information about the med-muscle ontogeny, constituting the first molecular base on the development of this economical and ecological key species. Thank you so much for your attention, and it's time to ask your questions.
Thank you. Okay, thank you very much, Marion. Someone wants the floor. Otherwise, I have some questions from Stéphane that I have to answer. So, François, perhaps, or Xavier, Marguerite.
Oui.
François.
Des questions?
Oui.
Belle présentation, Marion. Merci. Moi, ma première question, c'est sur quand tu nous parles du fait qu'il y a un tiers des gènes. Oui.
So could you switch in English because there is people that is supposed to follow us, and perhaps nobody can. Thank you.
Good job, Marion. Really nice presentation. My first question is going to be about the third genes. A third of the genes that you found were present or absent between individuals.
Yep.
And it sounds very surprising to me. Are you talking about present absence in the genome?
Yeah, in the genome.
Yep. At the transcript level.
Okay. I'm going to retrieve the good slide. Yes, there we are. So yes, it was really surprising. So in this paper from Gerdel in 2020, that was also surprising for them. So it's when they sequenced a genome from one adult, and by resequencing 14 different individuals from 2 other populations, it's the moment when they retrieve those 20,000 dispensable genes from a core set of 45,000. So it's really, really huge. And that's also the particularity and the difficulty, I would say, of this species. That's the reason why we had the idea of comparing the de novo and the genome-guided assemblies, because most of the time, when you have a species where the genome is known, you say, okay, I will do the genome-guided since it's more fast. In 1 week, it's done, and it's also more reliable because it's not limited by, for example, the short read length. But in this case, that was also the major objective of this study. It was to determine with the de novo if I was able to retrieve this individual variability and in which measure it could bring new information or disturb the potential analysis.
Whoa. Okay. I need to get my head around that because it's such a weird thing to like think about when you do sequencing and then use, like you said, a genome, a reference genome. If like you think of that as like maybe a third of my genes are going to be absent when I do my study, then that should be reflected in the results, you know. But I guess what you're doing, it's because you're working on a cohort, you don't have this problem. But if in the future you want to do sequencing on a single individual and have like replicates of that, like to think that different replicates can have like wildly different number of genes, that can be a real issue in your analysis. So it's very interesting. And that brings me to the next question, which is, how did you decide on the experimental design? I didn't really understand why you would have like 4 different pairs producing gametes or larvae, and then you pull the larvae and you only have 2 groups at the end.
Yeah, I will come to the slide. Just it's in material method just after. Up. Okay. I'm sorry. The material method.
It's very slow.
Voilà, just after. So I decided the reason why this design has been done. So the reason why we pooled the larval pool 1 and 2, the major reason is that because we were afraid not having enough material. Because the problem is that on each larval pool, you have to sample 15 times each. And the problem is that when you extract the RNA, if you don't have enough RNA, you cannot do anything with that. The further analysis cannot be performed. So the reason why we decided to pool those larval pool together, it was a question of the quantity of material to obtain. And finally, we could have done the larval pool separately. We could have done it, but it was one of the major difficulty. Like, what we do if we don't obtain enough material. The problem is that it's on a spawning season. It's really long to get there. So you do your spawning season, you sample, and finally, when you extract your RNA, you realize that you don't have enough material, and you cannot do anything with that. So it was the major reason we decided to pool.
But I mean, like those mussels are not an endangered species. You have tons of them on the rocks. Like, why would you not start with more individuals?
That's true. It's just that it depends on if you have time, if you want to address a particular question, if you have to get answers to your question just after the spawning season, you cannot spend time to do it again. But it can be something that can be done in the future for the next spawning season, for example, it can be done.
So you had no idea how much RNA you were going to be extracting from your samples?
Yeah, we had no idea.
Of time? Okay.
Yeah, we had no idea. It was really to be sure if something happened and to be sure to have enough material.
Okay. Can I ask another one? Paula? Yeah. Okay. So, when you compare the number of genes that you get from the de novo assembly to the genome guided?
Yes.
Yeah. Do you have an idea of how much of this is due to producing fragmented sequences, contigs?
I'm sorry, I didn't get your question.
So you get when you do a de novo assembly, you get like hundreds of thousands sequences, and your explanation for that was that maybe these genes are indispensable, so they are.
Yep.
But there's also the fact that when you do a de novo assembly, you produce a lot of fragmented sequences of sequences that are not overlapping, and you're not reconstructing the whole gene, right?
Yes, exactly.
So I wanted you to tell me if you have a feel for how much of the difference that you see between 60,000 and 128,000 is due to that, is due to incomplete assembly of the transcripts.
Yeah. So if I go in appendix, so basically I will just show you there. So during the BUSCO assembly assessment, there is it gives you also a score of the fragmented BUSCO, so the fragmented reads that they found in the analysis. And when I performed the BUSCO analysis, I had a really low fragmentation. And also, that's the reason why I decided to apply filters, because transdecoder, it will say that your transcripts are corresponding to a pool of known protein within the metazoans. So it's a way to check that the transcripts you found are kind of real, or in the case, they match with proteins that are known. And also the CD-HIT filter, it's filtering the isoforms. So that's also the reason why it was mandatory to perform a quality assessment with BUSCO and the two filtrations with transdecoder and CD-HIT. That was the major reason to be sure that what you observed, they are not reads that were fragmented and that have been assembled in a wrong way, let's say. I will try to find the annex where I have the BUSCO results. Yes. So here you have the percentage of the fragmented, you have the percentage of the fragmented that you obtain with the BOOSCO result. And finally, we obtain 0.73%. So it's really low, it's less than 1% that were fragmented. And then after that, you perform the transdecoder filter that gives you even more like certainty about the quality of the transcript you have.
So sorry if I can I jump with a question.
Okay.
So sorry for being late.
Thank you for
really apologize for that. At least I got the last part of your questioning. So maybe you already answered to the question, but just to follow up this question, did you play with the parameters of both of the transdecoder and the CD-IT? Because if you have a certain window where you can play, you may lose or underestimate. But considering the high number that you had, probably did you play around with these parameters?
Yeah, exactly. So, for example, for the CD-IT filtration, they say that the threshold, the classical threshold, is 90%, and I try to play with this parameter to get I did a CD-IT filtration at 99%. And I saw literally really the number that was changing a lot. If I go once again back, I will try to show you. But yes, I played with the CD-IT filter, and I saw that according to that, the number of transcripts really changed. And even after the transdecoder, there was a really high number of transcripts. And as you can see, if you filter with CD-IT at 90%, you are more stringent. You decrease the number of transcripts to 73, and with the filter at 99%, you have 120. So yes, I played with those parameters, and I tried to, because I had a discussion on. Of course, the result that you obtain, it depends on the method, on the threshold you choose to apply, and it has to be according to the literature. And I think that what is usually accepted is that you should have about like 2-3 times the number of transcripts compared to the number of genes, something like that.
Kind of a rule of thumb, yeah.
So I have to still play with this parameter, but I was wondering, how do I choose this filter, like only based on like let's try, and I will see the numbers I got. That was I think the best solution to try and to see.
Thank you, Marion. François, do you have another question? So no, we move to Stefano. Do you want to continue?
Well, I don't know if you. I gave you some. I gave to Paula some of the questions just in case I wasn't able to join. Which was almost the case. But yeah, I have. If there is time and you have a little bit of energy left, I had some questions. They're not mean. Don't worry.
Also,
I have just a technical question, because which internal control did you use for your delta CT calculation for the qPCR? Because I didn't. Maybe I missed it, but I didn't find it in the manuscript.
Yeah, you mean the housekeeping gene that I used for the qPCR?
Yes, exactly. Yeah,
exactly. So based on the literature, I used elongation factor. So there are some papers that are contradictory between each other, saying that sometimes elongation factor doesn't have a constant expression over time. But I checked on the in silico, thanks to the in silico analysis, both with the genome guided and with the de novo. And finally, I had an expression that was stable from the 0 to the 72 hours post-fertilization. So I really had this control, and also the literature supporting that sometimes it was used as a housekeeping gene. And so I was like, okay, I can use it as a reference.
So you just went with one, you didn't try more than one, just to be on the safe side?
I followed what they said in the literature, and also in my in silico analysis, I saw the profile that was really stable over time, and that's what we want for a housekeeping gene.
And I have staying on the technical side, you use for the in situs, you use the HCR protocol, right? Which is a pretty cool way to do in situs. I personally never used, but I was reading, and sometimes you have I mean, did you do, or someone else did, some control with the normal in situs with the normal in situs protocol to see if you can mimic, if you don't underestimate, or maybe more likely you overestimate because you're amplifying. Sometimes there may be this chance. So for some.
Of course, yeah, a negative control has to be performed. It's just that in a 6-month internship, so it was really quick. I did all the bioinformatical analysis, so I didn't have the time to do the negative controls. But of course, because we are developing this technique in the lab since about 6 months, and of course we have to perform negative control to see if this staining are specific to the mRNAs, and also to look at the potential autofluorescence of the muscles that has already been observed in the literature.
Okay, I mean, I have to choose because I have a few questions, because I really enjoyed reading your manuscript, and so there are of course several questions that maybe we will discuss even another time. But I have a couple. So did you maybe you already answered? Did you try? So it's pretty exciting, these pan-genomes and this amount of genes that are species-specific of transspecific. A general curiosity: is there any other metazoans that has such a feature, and that it's reported that in such a heavy way? Because
that's an excellent question. I didn't find any literature information about another, because we say when they have a high individual variability like that, we say an open pan-genome. And that was the only species where I saw that. Maybe I should have a look to if there is another metazoan, but I didn't have a look to. I didn't find another species.
So when you do this, when you do the de novo assembling, right? So you basically pick up all these what you call dispensable genes that are basically cloud, or I think it's probably more cloud or shell genes. Yeah. So I'm interested in what is the nature of this bunch of genes. This is if you know anything about it, or if there is something about this. And the other related question is, if you filter out these genes from your de novo assembling, do you have a more comparable heat map, comparable to the genomic based assembling, or do you think you have it, or did you try, or
I think about it? The major problem is that, so I tried before the filtration. I tried to generate the heat map. I think it was just after transdecoder, but before CD8 filtration. The major problem is that I had 1 million, near than 2 million lines. And on R, if you try to generate a heat map with 2 million lines, it takes you the day. And so I tried it once, and I obtained almost the same clustering, the same heat map than the one generated after the CD hits.
Okay.
But after the discussion with the bioinformatician, he said, no, you have to filter everything, and that's no way to perform such analysis on 2 million transcripts. You have to filter.
Okay.
Stefano, we have some minutes left, so perhaps we go around and we come back to you if we have time.
Sorry. Didn't want to monopolize. Thank you.
Xavier, do you have some questions?
Well, most of my questions would overlap the ones that has been asked by my colleagues. Maybe one thing regarding the life cycle of the muscle. What kind of, is it an easy job, or did you observe a lot of mortality, or how, tell me. No, okay, I can maybe precise a little bit. from my experience, trying to complete life cycles of many marine organisms, I often met issues when you have abundant gametes, sperm, and oocytes, and you observe very quickly Morilla, Blastula, Gastrula, but sometimes you really observe high mortality levels or rates. And I was wondering how this, how you whether you manage to avoid such mortality or.
I think it's almost the same for all the species. Like in the laboratory, you never know. Like you do the fertilization, and then you observe your larvae growing. And of course, I observed mortality. The problem is that for the iron extraction, we don't feed them because it's better for the RNA quality and to not having contamination by feeding them. But of course, if you have mortality, that's just you never know. It's just like the common life of the lab. When you have a species, it's developing well, and if not, you restart your experiment. So with the mussels, I think it's like with all the species. For example, actually, it's the spawning season of the Antioxus, and we are also observing the same with a lot of mortality sometimes in the plates. So I think for the mussel, it's exactly like the other species. You can have mortality, and it's what is really hard to work with living organisms at the lab. That's the lab life.
Actually, this is a key aspect for me because completing a life cycle means involving also a lot of efforts in order to understand the best conditions, to be sure to observe survivals. Then of course you can be sure that the samples you will realize will be of a good quality. Could you imagine some improvement in order to have better survivals or less mortality?
Yes, mortality. I mean, for example, I think one major key will be the quality of the gametes, like to choose the adults that seem to be the more with the sexual maturity, that are more in good health. And when you see that the gametes are not in so good health, you decided not to fertilize it. I think the one way to play on it will be to choose the best adults to fertilize, to do the fertilization in the choice of the gametes. But I mean, we select some adults, we do the heat shock, and after we try, and if it doesn't work, we restart it again the day after, and it's trying and trying. But that's a good question, how to improve the surviving. But I mean,
it's important regarding, according to me, but again, it's not a critique in the sense, it's just a discussion. It's important to keep in mind that if you really want to have access of a good transcriptome, you can also easily imagine that if the condition of rearing or of the culture are not ideal or good, you could imagine that you have a kind of underestimation of the quality of the transcriptome you will obtain from the different samples you will operate during the ontogenesis, or at least the early embryogenesis. It is, for me, it's a crucial point to pay attention to this aspect, as completing life cycle is a very complex step. Otherwise, I wanted to tell you that your demonstration was of a great quality, and again, congratulations for the work you performed. I would have more questions, but I clearly understood that Paola asked me not to be talkative and more talkative. That's clear.
Many thanks. So sorry, Xavier, but I would like to leave the floor for one question, one last question to Margarida.
Thanks for your presentation. Very clear. It's. I cannot do any technical question or remark about the protocol because this field is super far away of what I'm doing and what I'm actually doing. And you already answered so many questions. So, but I have maybe curiosity or when it could be interesting to do this de novo protocol, when it could be interesting to put all this effort to know the individual variability of the muscle.
This individual variability and the isoforms that are retrieved with the de novo could participate, for example, to link the muscles adaptations to the environment and because the individual variation, it could be also adaptations to the local environments. So to link the muscle with its direct environment and the potential local adaptations in the different sites could be reflected by this de novo additional informations.
So this maybe has a lot of aquaculture interest also.
Yeah, in applications, yes. Could have.
And another last curiosity. Sorry.
No, go ahead, Mark. Go ahead.
And I have a curiosity because sometimes I'm cultivating also algae, super different. But during these hours after fertilization, how you keep the larvae? In which conditions you keep them?
Yeah, in which condition you mean?
Temperature or in a fridge with light, without light, filtered water, I don't know.
Yeah, we try to keep them at, I think it was 18 degrees in the temperature. So we put them in an incubator at 18 degrees and in filtered seawater in metal plates.
Thank you. That's all.
Many thanks, Marion. Okay, I will not have any question because there is so many aspects. I have ones that perhaps you can answer in one word, but no more. Is there some risk that the reference genome that you use is not a pure Mytilus galloprovincialis genome, but it could come from some hybridization, as I know that in this organism you can have some hybrids, and this could explain this discrepancy on the score and on the existence of undisposable genes.
Mm-hmm. That's true. It could be, I think, because in the genome study, the study of 2020, like when they have, because they have sequenced one female, so the reference genome has been constructed based on one Spanish female. So I assume, I don't know how they tested, it was really the good species, but I assume they are sure that it's really the good species. And it's like the individual variability, it was found by resequencing 14 other individuals. So I guess normally this species, the Mediterranean blue mussel, is it can be recognized based on because it's bigger than the common mussel, for example. It has the bluish color of the shell. So I think there is still a risk that it's not really this good species and they could be hybrid. It's a risk, but I think that a lot of precautions are taken, but there is still a risk, but I think they took all the precautions to be sure that it was really the good species before sequencing it.
Yeah, definitely. If they compare to 14 others individuals, I think it's the best answer that you can have. Okay. Yeah. Thank you, Marion. I think that we can stop here this long defense. And congratulations, Marion, and we will come back in 5 minutes with the next one.
Thank you, Marion. Thank you.
Nice YouTube's comments.
Thank you, Marion.
Second part of the presentation, right? Thank you. So let's go, Charlotte, when you want.
Good afternoon, everybody. My name is Charlotte, and in my internship in a company called Ecological on artificial habitat, an effective tool for ecological restoration in aquatic environments. Now we'll be following a scientific approach with first an introduction, then a method and method, and talk about the result in discussion, and finish with a conclusion. The constellation phenomenon is due to a rapid increase in the population, which has a marine and freshwater ecosystem experiencing various anthropogenic pressures due to the constant human activities, such as agriculture, tourism, pollution, and construction of infrastructure. This urbanization has led to habitat destruction, which is a threat to aquatic biodiversity. The freshwater littoral harbors an important biodiversity and provides various habitats, such as nursery, feeding, refuge, and breeding habitat. But habitat destruction is due from different causes, such as the construction of dams, which lead to a drawdown phenomenon, and the artificialization of littoral. Going to the threat in the marine environment, coastal marine fish species depend on coastal ecosystem as they provide nursery habitat, which is essential during their natural life cycle. But the construction of harbors and seawall destroys its natural habitats, which then discontinue the natural life cycle, then leads to a mortality rate close to 100% for the new recruits. Therefore, habitat destruction in both ecosystems leads to serious consequences on biodiversity life cycle. So how can we limit the effects of habitat destruction? Well, we can use ecological restoration. I'll be giving a large-scale definition of this term. Ecological restoration is the process of assisting the recovery of an ecosystem, where the aim is to repair the structure, the functioning, the composition, and the services of the targeted ecosystem. Ecosystem offers two habitat restoration solutions. In freshwater environments, we'll find the floating ecosystem. In the marine environment, we'll find the biota. Both solutions aim mainly to restore the nursery habitat. Therefore, the objective of my thesis was to see these artificial habitats are an effective tool for ecological restoration in aquatic environments. I will be able to answer this question thanks to the observation of two emblematic and keystone species. In the marine environment, we will have the sea beavers. In the freshwater environment, we will have the northern pike. For the material method, I will start within the marine environment with the diode. I will be presenting two designs of the GAN, the pontoon and the dock diode. These two designs are grid cages with two liquid compartments. One of the compartments is filled with a local substrate, which is usually oyster shells, providing a habitat in a feeding habitat. On the side, we have 1 to 2 empty cages, compartments, allowing the protection from the predators. This diode has been deployed in ports worldwide since 2013, but for my thesis, I was only interested in 51 equipped ports along the French Mediterranean coast. The surveys are conducted following an underwater visual sensors protocol, where 1 to 2 certified divers will go and identify the fish species and their abundance within the diode itself and within one perimeter of 1 meter around the structure. The surveys are conducted once to twice a year per port. Going in the freshwater environment, we will find the floating ecosystem solution, such as the artificial floating littoral zone, hereby named as SWIM. It's a 70 square meter structure with two parts, a terrestrial part and an aquatic part. On a terrestrial part, instead of having terrestrial plants, we incorporated hydroponics. The aquatic part is a 48 square meter area with 3 different zones. On the side, we will have 2 shallow decks, allowing with grassy vegetation, favoring the reproduction of phytophilic species such as the northern pike. The central deck is a deeper zone with larger microcliffs. These 3 zones allows microfauna to have access to vegetation regardless of the water level in the reservoir. We also had incorporated around these decks another design of the BOS, allowing the inside of the structure to be more sheltered and protected from the outside. Therefore, 3 fleets were deployed in Serpent-Son Reservoir, which is located in Provence-Alpes-Côte d'Azur region in France. And this reservoir gathers water from the Durance and Ube rivers. The surveys are also following an underwater visual scientific protocol, where 2 certified divers will swim or snorkel around and within the structure to identify the species and their abundance. Following this protocol, 12 surveys were conducted over 3 consecutive years. As a long-running project, the data from my thesis was collected and gathered within the ecosystem database, within scientific publications such as thesis and grade literature. Over 300 marine species were observed in the biota since 2013 worldwide, but as I mentioned earlier, I'm only interested in one species, the dusty grouper Ecological Restoration. They have been observed in the biota since 2013 along the French Marion coast, but I'm only interested in the observation between 2016 and 2021. Eighteen individuals of dusty groupers were sighted at 10 different locations. Between 2016 and 2020, we had observed between 1 and 3 individuals per year. In 2018, both individuals were sighted on one day in one location, which is in Corsica. We can see there's an increase in individuals observed in 2021 with 9 dusty groupers. However, this increase could be biased to the observation of 4 individuals sighted in one day in one location, which is in Bulgaria. In addition to these dusty groupers, we had one rare observation of a multi-grouper in Port Canto, Canada. Out of 18 dusty groupers observed, 5 of them were sighted at an unusual area, which is in Port Gardien. It is unusual as this port has sunny bottom, whereas the dusty groupers do prefer rocky habitats. So what does this situation entail for these 5 individuals? Will they die, or will they be able to survive once they reach their refuge sites? Will they be able to find a suitable habitat within the surrounding of this port, and therefore participate in the renewal of the species population? Or, as I mentioned earlier, for this observation was sighted on the same day, is this port just a resting area as I migrate towards a suitable habitat further along the coast? However, the increase in the dusty grouper population along the French Marion coast is due to 3 main factors. The first one is the partial protection due to the implementation of the moratorium since 1993, which reduces the anthropogenic pressure of overfishing. This species has benefited from this moratorium as it's approaching a set glacial equilibrium, favoring the female. And lastly, this species do benefit from climate change as they prefer warm water. And the water temperature along the French Marion coast is increasing, favoring their return. Therefore, the biota is a contribution to the species increase in population, as they serve as nursery habitat, restoring the natural life cycle of the species. Jumping into the freshwater environment, we observed 25 individuals of marine life between the size of 20 and 250 millimeters. The observation were only sighted during 6 surveys out of the 12 conducted, and exclusively in late spring and early summer. This time frame is quite late to observe young life, but this is due to the water temperature in the reservoir. Out of the 25 individuals sighted, 20 of them were larvae with a size of 30 to 30 millimeters. These 20 larvae represent 80% of the observation of marine life. Out of the 20 larvae, 14 of them were observed in one day in one of the 3 structures, which is the highest density of prey observed in this model. And lastly, the large angel is a 250mm subadult. If that was observed in the police, it either takes refuge as the resting or shelter area, or it can feed on the suckling that was present. Despite the presence of grassy vegetation, microphytes, and hydrophytes, no spawning of the pipe was observed during the project. The typical reason given by scientific papers is that the eggs are quite difficult to observe, as they're small, or approximately 2 millimeters. A female do spawn over a large area. And the last reason is that the structure is quite far from the shore. The distance between both shore and the structure when the water level is the highest is 50 meters, and there's quite a depth. And there's no habitat in between. However, it's thought that you should have some potential spawning, as we observe pipe larvae, and it is known that these larvae do face themselves following mutation. And as the distance between the shore and the police is quite large, the first instinct of these larvae is not to swim across the reservoir to find habitat, but to fix themselves on vegetation. Furthermore, in other projects conducted by Ecological, we observed two angels spawning, breeding. These photos are taken by a camera feed. We also observed one pipe egg among the 1,000 that were spawned in the area. Therefore, the larvae observed with the yolk sac still present are believed to be born on these structures. Therefore, there's a high probability of spawning in the past, and there's going to be one in the future on these structures, as the Northern pipe has a homing behavior. This means that the pipe angel likes to reproduce on an area or in a habitat where they're born or grew up. Therefore, with this behavior, there's going to be a long-term effect on creating habitat for this structure. Thus, further surveys should be conducted in the next few years to observe this effect. Thus, the police provide various habitats, such as the refuge and habitat for larvae, juvenile, and adult freshwater species, a feeding habitat with the presence of saprophytes larvae, and a spawning habitat that has been validated with the presence of freshwater greenies eggs that were in the oyster shells present in the structure. As a conclusion, the biot is a simple, efficient technology solution, whereas the police is a larger, more complex one, and is larger and more material. Both solutions provide ecological function of nursery. However, the biot and police are not ecological restoration tools, but more ecological function of rehabilitation tools. Furthermore, these tools are becoming, these structures are becoming scientific monitoring tools. During my internship, I had a chance to touch the different aspects, such as participating in scientific monitoring, helping the communication department, where I designed a communication panel for a project, where I created a freshwater game, and participated in different events. But the biggest core of my thesis was to write 2 short communication notes on the observation of dusty bloopers and the observation of northern pipe within the police. I also had published a blog article in one of the biggest ecological restoration notebooks on the ecological restoration operation of the French Mediterranean coast to promote the dusty blooper recovery. I would like to thank Sebastian and you for being my supervisors for these 6 months. They helped me, guided me, and supported me during the whole internship. And I would like to thank the whole Ecosystem team for allowing me to participate in different projects and aspects of the company, and for the amazing atmosphere they did at the office. Thank you for listening.
Thank you, Charlie.
Thank you very much, Charlie. So we can start with the questions. Margarita, perhaps if you want to start.
Thank you. Thank you very much for the presentation. And congratulations, because you did many divulgation and a lot of activities, scientific publications, and a lot of valorization of your work. And first of all, well, these structures, I guess they work increasing the structural complexity of the habitat. They create an habitat suitable for the species to proliferate. And you say that this could limit the effects of habitat destruction, because which habitats do you think which habitats they replace, which functions they incorporate, which habitats have been destroyed, do you think? Which are the natural nurseries that should be present where this
The fjord one or
like for example macroalgae, which natural habits create that complexity? Or maybe I'm not explaining well.
I didn't quite understand the question.
Like the habitat that has been destroyed, like when you construct a harbor, for example, it's can be different types of habitats. Can be like rocky habitats that create a 3D complexity, or macroalgal habitats, or Posidonia meadows, maybe. They try to replicate the functions of these habitats, these artificial.
So the oyster shells is a subject that kind of replaces the rocky habitats, and all the structures, as you can see, there are some falling with a lot of algae. So some algae habitats are also incorporated in the structure. Was that the question?
Yeah, perfect. And there's some limitation to put these habitats. That we should put them on with some limitations for fishing, for example, or it's better to put them in protected areas? Do you think their functions could be increased if we, depending on the place where we put them?
Could be. I don't think they're implemented in protected areas already, but in protected areas there are like already less economic pressures, and I think the aim of the company was really to put them in ports where the habitats are really the most destroyed, helping as the larvae are really like searching for habitats, and they're entering like ports and they're finding no habitats, and they're like have a bigger mortality rate in this area. So I think the objective is really to like help the general survival within the ports.
Okay.
And not because in protected areas are already kind of protected most of the time. Mm-hmm. So it's really like trying to find another area to
where are more urbanized areas or harbors or ports.
Yes.
Thank you very much.
Thank you. Who wants to ask Stefano, François, Xavier? François.
Yep, okay.
Open your microphone.
Good job, Charlotte. My question is about the, like the numbers that you show for the two species, they seem quite low. And so I was wondering if you looked at different species, other species, and they were low for these two specific species because they're like, bigger predators. And
so for the dusty groupers, they're increasing numbers, but they're still low. But it's only the numbers seen within the structure and not around it. So I don't know the numbers of around the structure, like in other habitats with no US, but they're still low compared to like the SAR. We quite seen them quite a lot. Pretty much at every survey, we at least see 1 to 2 individuals. And for the northern pike, the 25 individuals are only for the underwater visual census protocol, And there are other protocols that were studied that showed more numbers of individuals. There's I think there's like fishing, the card fishing, which is another system of capture, and I think the NAS also. They're like different protocols for, and where they also see young pipes in them.
But my question was about like, did you see different? Did you look at other species than these two? And if there was like more of other species?
There were for my thesis, I only looked for these two species.
Okay.
Perhaps, François, just I interrupt you because I have a complementary questions. Is okay? Did you look just to those species, but did you also have some other qualitative info around those species? For example, male, female, or the size of the individuals. I don't know if it's easy to have a year or an age of those organisms, or you know what I mean? Do you have some other qualitative data sets?
Yes. When we survey, during the survey, I just showed the number of individuals. But during the surveys, we noted the size, mostly like to see how big they were and to see if they. And over the years, I think that once one individual in 12 months was seen several times over 3 or 4 years, still growing. And female and male, it depends on the species and how big they were to be able to see. And yeah, for the Nordic pipe, the same. We took the size, female or male, if it was possible, if it was an adult or not, and stuff like that. But not quite more.
Thank you, François.
Yeah, Charlotte, I'm going to bounce back and try to get you another chance to answer Margarida's question, because I was very interested in that question. And I'm going to try to reformulate it for you to understand. She was talking about, those rafts, for example, the structure, the artificial structure that were used. What kind of natural habitat were they mimicking? Do you understand? Like what kind of habitat they were trying to?
They're trying to mimic the littoral habitat. Really like the shoreline habitat of a reservoir, with vegetation, rocky parts of the habitat, vegetation habitats, rocky habitats, mostly.
And for the lake, because I like this floating raft on the lake. It's not something that
it's really a floating littoral structure, which like follows the water level. So regardless of the water level in the reservoir, the microfauna can have access to a grassy vegetation or to rocky habitats.
Okay.
So it's really like a littoral zone structure that's following. Wow.
Cool. Thank you.
Thank you. Xavier or Stefano?
Well, maybe I have a remark or a kind of question. I can imagine that it's not an easy task to mimic or to biomimic an environment that has been destroyed or at least highly impacted by the human activities and so on. And I can imagine how difficult it is, and for that I admire a lot this kind of works, and because I can measure how difficult it is and what is the challenge behind. But I'd like just to ask you something. In an environment that has been already impacted, when you install all these structures where you try to reconstruct a kind or any levels of actions, a kind of multi-trophic habitat, what are the chances to bypass the anthropogenic pressure acting at the place where you install the structure? In other words, do you think it is realistic to install structures in order to restore habitats in places where there is already a human pressure?
Well,
it's not my question is not a trap. It's just a kind of curiosity.
For the biote in the marine environment, we cannot really reduce the pressure of boating since it's like they're installed in ports. So that pressure you still have it. It's just to incorporate in ports to have a nursery habitat for the larvae. And for the floating ecosystem in the freshwater environment, since it's a structure that is anchored, I think you cannot. There's a certain distance you're not allowed to go within with a boat, but it doesn't really reduce the anthropogenic pressure around.
Okay.
Thank you, Stefano.
Yeah, thanks, Charlotte, for the presentation. It's not my area of expertise at all, so I just have some questions that may sound naive, but you present a lot of points where you collect your measurements and where these structures are deployed. So what are all these different habitat, the original habitat, homogeneous in terms of, for example, abiotic parameters, salinity and temperature fluctuation in the year, or are they comparables? And if they're not, did you see differences in recruitment and in your structures in the different spot? Maybe you show that, and maybe I've missed. So I apologize if I did.
Do you mean the different location of where we installed the biote on the French coast?
Yeah, you have several locations, right? So and I assume that in one there is maybe a river, so the salinity it's different. In the other, it's an etang, so there is more difference in temperature during the year. So do you have different results? Remarkably different results in the different points, or not?
I'm trying to look back at what I saw in the database. I think there's different results. For example, there are biotes installed in Valgaras, which is a port that actually is where river is actually going into the sea, and so the salinity is less, and it has a bit less species, but less abundance for sure. I think it's different species. But depending on where you are along the coast, the abundance is different, and I think some species are pretty much seen pretty much everywhere, and some others are not.
Okay, so then I had another question. So do you have maybe it's overlap a little bit with the question of Francois and Paula. Do you have a record of how was the situation before the certain disruptions of the different local environments? So in other words, your recruitment and your analysis, are they mimicking in some way what was the situation before, or is drastically different still with the recruitment of these species that you look? You see what I mean?
Yes. For just the Dusty Cooper, or in the Northern Pike, or for all?
Yeah, it's basically in before and now. You're looking and you present the data of the now. Do you have some data in the records of?
I don't know. We had data about in the sport before the implementation in 2013. I believe we do, but I do know at that moment there's a project called HISPIA, with the servants network of stage recruitment, following the special temporal of the larvae recruitment. And there are different zones along the French coast that are looking at, and not only looking within the field, but also at, on natural habitats. And I don't know the result of that, but they're looking at both. And I think there's some difference, but not 100% sure.
Okay, thanks. I have just one last question if I have time. So you look through the, there is an observation through the 3 years. So I suppose when the structures are deployed and at the end of the last observation, you built up an ecosystem. So the one that is at the beginning, the one that is after 1 year, and the one that is at the end is not different. And did you or your colleagues monitor a drastic change? Because it's an ecosystem that is built from basically from scratch, if I understood. So do you see some way a development or a change?
There's a change for the build, because when you install the build, it's like it just steel with oyster shells. And over the years, I think the first few months there are not much fish species coming in. Where there are the algae are coming, they're like pulling the structure, and then once the algae are there, I think crabs and shrimps are coming, which then make like kind of a lot of noises for the tuna or for the larvae fishes that orient themselves to these habitats to have nursery habitat. So I think furthermore, in the years you have more evidence of different species, but I think at one point after 2 or 3 years we have quite the same species.
Okay.
And for the fleas, I think it's the same. It's like for northern pike, they're like starting coming. They went there this year. They went back. They saw pike larvae again. So in the next few years, they're going to be coming back and probably be more individuals. Who knows? But that's the process of going further.
Thank you very much.
Thank you, Charlie. So just one last question from me, because you already have many questions around. And I'm sorry, but again, I come back on your data set. And my questions, it's more linked to your conclusions. And I didn't read your manuscript, and your presentation didn't tell me how you really evaluate your work. And it comes back to a question that Stefano just asked you, and how you can say that your dispositive is really increase the amount of those organisms. What is your kind of analysis? What kind of statistic you did? What you compare exactly in between? Do you have the wildlife and your dispositive, and then you compare both?
So I know for the northern pike, from what I understood from the there's a comparative between, there's a study. So I only looked for my thesis on the structure, but the doctorate who did the whole project looked at the littoral zone, the natural habitats, and on the fleas. And if I remember correctly, there was more juvenile on the fleas than on the littoral zone. And for on fresh for the marine environment, I cannot answer that question because I don't know the answer.
But for example, Charlie, when you say that you have this increase on groupers, for example, how did you test it, the increase? Okay, we saw that you have value that it increased, but how significant are those values?
I didn't do any tests, nothing. I just, with the observation seen between the years, you say, oh, there's an increase. But as I said, the increase could be biased because there's only once or twice, the surveys are conducted once or twice a year. they're living, they're mobile species, so they move a lot. So we can go one day, but the next day it could be completely different. So I cannot say 100% there's an increase, because there's also, we're seeing today more juvenile dust keepers and more dust keepers, because there's also, compared to 20 years ago, because there's more surveys are being conducted today along the coast. But that's why I say that the biota is only a contribution to the increase in the species population.
Okay, thank you. Yeah, it's my answer. Thank you. Thank you, Charlie. So we can stop here now. Thanks you very much. Thank you.
We are back. Paola, can you hear us? It's okay. Agata, can you try just to say a few words?
Yeah, good morning, everybody. I can't see you from the screen, but I hope you can hear me properly.
You can
Yes,
see them.
if you turn back, you can see me. Hello.
Sorry.
We cannot see us, but we can see you.
Perfect. Tell me when I can start.
When you want.
Okay, perfect. So welcome everybody to my master thesis presentation on the role of Wnt beta-catenin pathway in regenerative processes in the sea anemone Nematostella vectensis. My internship was under the direct supervision of Dr. Dina Miao. To give you a little bit of context, I did my whole internship within the Eric Rottinger's team in the stress response, regeneration, and aging team within the Institute for Research on Cancer and Aging in Nice. And my internship was focused only on the regeneration part. But what is regeneration? So according to the National Institute of General Medical Sciences, the regeneration is the ability of an organism to restore damaged tissues, missing cells, organs, or the whole body. These processes has intrigued scientists for at least 2 centuries, but humankind have been fascinated by this process for millennia. You might know the myth of Prometheus, where the Titan was cursed by Zeus with an eagle eating its liver for eternity. So liver and eternity are not randomly put in the story, because liver is one of the major organs that can regenerate in the human body. And already back in the day, Greeks knew that. So as I previously mentioned, regeneration can be occurs at different levels. At cellular level, most of the organisms can do that. Tissues and organs, humans are within this kind of species that can regenerate tissues and organs, as I previously said with the liver. But also zebrafish, for example, can regenerate part of its heart. And there's a last category which can do whole body regeneration. These organisms are very rare in the Metazoan kingdom. Also, regeneration is very complex, and it's a network of molecular cascades that are involved for making regeneration possible. And during my internship, I was focusing on one key molecular pathway, which is the Wnt pathway. But how does it work? So first, we have some Wnt ligands that are secreted outside the cells that bind with the freezer receptors, which is on the level of the membrane. And when they bind together, the freezer receptors are activated and allow the block of the beta-catenin destructive complex, so that the beta-catenin can accumulate in the cytosol and further translocate from the cytosol to the nucleus. And last step, the beta-catenin, once in the nucleus, can act as a co-transcriptor factor, starting all the process of regeneration. The Wnt pathway has the particularity to be high conserved among animals, and it's very widespread among organisms. Here are some examples. First, in mice, is involved in skeletal muscle and pancreatic cell growth, but also in zebrafish is involved in tail regeneration, for examples. And for amphibian axolotl is involved in limb regeneration. This pathway is also found in invertebrate. Here you have an example of the planarian, where the Wnt is involved in the arboreal regeneration of the animals, and also in Hydra vulgaris, where wind is involved in head regeneration and also cell proliferation. So, for my internship, I wanted to study this pathway in a non-conventional biological model, which is Nematostella vectensis. But why using a sea anemone rather than a mice or a zebrafish that are more conventional as models? So first, we have a whole body regeneration abilities. The sea anemone belongs to the lucky category that can regenerate the whole body. Here in the picture, you can see the staging system of head regeneration in Nematostella. Then it's very easy to handle because the adult specimen have a length between 1 up to 3 centimeters. And then it has a controlled life cycle in the lab, meaning that we always have stock for this model. It has a translucent body. In fact, you can see that if we need some experiment, we can clearly see the pharynx on the arboreal part or the mesenteries. And finally, we have a full sequenced genome. So we know already something on the wind pathway in Nematostella, which will be very useful for my biological questions. First, the wind pathway members have been listed, and most of the vertebrate wind genes have been reported in Nematostella, except the wind9. And also, we have the list of the frizzle receptors. Then, some work on my lab, previously to my internship, already detect the expression of wind ligands and frizzle receptors during regeneration. And third, it has been reported that wind is necessary in the oral fate and axial patterning in Nematostella. But we don't really know in regeneration when and if it's sufficient, the wind pathway. And here comes my biological question. Is the wind pathway required and sufficient during regeneration in Nematostella? To tackle this question, we used 3 different sets of experiments. First, we wanted to assess the regeneration success when we inhibit the wind pathway in Nematostella. To do so, we incubate the animals for 7 days with 2 different wind inhibitors, EndoIWR and ICRT14, to assess the regeneration rate. We also know that regeneration and cell proliferation are linked in Nematostella. So we also wanted to see whether when we block wind, the cell proliferation is also blocked. The protocol was kind of the same of the regeneration assay, but in this case, we incubate the animals only for 48 hours to assess the number of dividing cells with an EDU pulse. And finally, we wanted to have a larger scale of the role of wind, and we wanted also to see the connection with the other pathway. And to do so, we wanted to see the interplay with MAP kinase. And in this last experiment, we incubate the specimen independently with the 2 different inhibitors for the wind pathway and also for the MAP pathway. So some words about the drugs I use, because it's very important to understand. We use 2 different inhibitors for the wind pathway, it's true, but their site of action is different. So we wanted to see how the animal responds to that. The first inhibitor, EndoIWR, acts at the cytosolic level here, which actually stabilizes the beta-catenin destructive complex. Whereas the second one, ICRT14, blocks the transcriptor factor in the nuclei, so no gene transcriptor is possible afterwards. And the interesting thing that we obtained were also in line with the previous literature about regeneration and the correlation with the cell division. So after 7 days, we obtained different phenotypic response by the animals. Here below, you can see 4 different phenotypes that we obtained, and these are not regeneration steps, but rather the response of the animal to the drugs. we obtain either a complete regeneration that was very similar to the control. In other cases, we had tentacles or pharynx delayed. More dramatic responses were when we had both tentacles and pharynx delayed, and in extreme case, we had no pharynx and no tentacles after 7 days. Our qualitative analyses show that if we compare the two types of drugs for endo-IWR after 7 days, still the most of the samples could reach complete regeneration. In fact, you can see that if control had 88% of full regeneration, with mounting concentration of drug, we still had 53% of the samples that could fully regenerate. Whereas when we were using ICRT14, already at the lowest concentration of drugs, 10 micromolar, we could see that 53% of the samples could not fully regenerate. We didn't observe pharynx and or tentacles. So we wanted to see more, and we wanted to see what was the effect when we inhibit Wnt on cell proliferation. And results actually confirm already what we obtained with the regeneration assay. In fact, if the number of cells wasn't significantly different with endo-IWR from the control compared to the drug treatments, with ICRT14, we could see a dramatic drop in the dividing cells already at 10 micromolar. And also for the interplay within the two different pathways, the untreated samples, which was our positive control, showed an expression of MAP-ERG. You can see it here. We have the expression. Our negative control was an inhibition of the MAP pathway, and 100% of the samples did not show any MAP expression. Whereas with ICRT14, which is an inhibitor of the Wnt pathway, we still have some expression of the MAP kinase. And here a graph showing that for the MAP kinase inhibition, we had 100% of MAP inactivated. When we inhibit the Wnt pathway, we still have more than 40% of the samples that have this pathway activated, suggesting that this pathway might act prior to Wnt. So we had a bunch of results concerning regeneration, cell proliferation, and also the interplay with different pathways. But what our results really tell about the Wnt pathway so far? So coming back to our biological question, is Wnt pathway required and sufficient for regeneration in Nematostella? Could we really say that it's required and sufficient? So first, we could observe that we have a phenotypic response, but the regeneration is only partially blocked or delayed. And also, very important, the phenotypic response depends on the drug we used, meaning that the site of action of the drug is very important. The animal might bypass when we use inhibitors that act at the cytosolic level. Second, we could say that the inhibition of Wnt pathway affect the latest stages of regeneration that are also the stages that are dependent on cell proliferation. In fact, we could see that when we inhibit the Wnt pathway, the cell proliferation was also blocked. And in fact, in most of the cases, we couldn't see the elongation or also the tentacle buds. And lastly, the space of the Wnt pathway, when we also assess other pathway, suggests that the Wnt pathway acts prior to the MAP kinase. So in a nutshell, the Wnt pathway is not the only major player involved in regeneration, but it's rather part of a complex network of molecular cascades that needs a logic temporal precise timing for that. Even though our biological question was partially answered, we still need to go further. And to do so, the next step will be to see whether it's truly sufficient, the Wnt pathway during regeneration. And to do so, we're gonna do some experiments by overexpressing the pathway in areas of the animal that usually do not regenerate. For example, here you can see by the picture a zoom on the epithelium of Nematostella deprived with mesenteries. And also another experiment to carry will be to validate the transgenic line beta-catenin M-cherry in order to assess the in vivo dynamics of the beta-catenin. In other words, to see the translocation and the moment when the beta-catenin translocates from the cytosol to the nucleus. So I wanted now to leave you with a quote that Richard Goss wrote back in 1969. If there were no regeneration, there would be no life. If everything regenerated, there would be no death. All organisms exist between these two themes. But is it actually the case for Nematostella? Thank you very much for listening, for your attention, and a special thanks to all the IRCAN lab team. A special thanks to Aldine that supervised me over these 6 months, and to Eric to host me in his team. And thank you all for listening, and to the jury back home. Thank you.
Thank you, Agata. So who wants to start? François, Stefano, Margherita. I can also start with a very simple questions. Agata, I would like to know, obviously it was about the specificity with the inhibitors. I'm just thinking that how do you fix the range of the inhibitors? The concentration range.
You mean the range of concentration?
Exactly.
Yeah, we used this range of concentration by first, there were previous experiments did in the lab, and also there were mounting concentration done on embryogenesis of the Nematostella. So we actually used a range between 10 and 25 because we could see that the tissue were not damaged after 7 days. So
thank you, Agata. And I have just another question that is still linked with these inhibitors. The fact that one has a stronger effect than the other one. Does it mean that the transcriptive factor that could be inhibited can act also without the catenin?
That's a very interesting question. I'm not sure because the beta-catenin is still a co-transcriptor factor, so it binds with the TCF. So I think there's still an action. It's not, I think if I get correctly your question, I think the animals when we use the weakest inhibitor, so to speak, it is because of the location, meaning that by stabilizing the beta-catenin destructive complex, maybe there's still some beta-catenin bypassing through. And an interesting experiment to carry about that would be to follow using the transgenic line. Would be interesting actually to follow the in vivo inhibition of the beta-catenin, meaning that treating like with endo-IWR the specimen and see whether there's still some beta-catenin expressed in the nucleus or not even in the cytosol. And so for ICRT14. That would be, I think, the only way to actually assess that it's not the beta-catenin the main actor, but also it's essential to bind to the transcription factor.
Thank you, Agata.
No problem.
I leave the floor to François.
Agata, what would be how did you, well, okay, so you chose these concentrations and you told us that the concentrations were based on like previous results, but how did you make sure, is there a way to make sure that the higher concentration that you used completely inhibited the pathway?
It's an interesting point, and I think to be sure about that we actually blocking the regeneration, not just delaying, would be to do incubation that are longer. For example, instead of 7 days, increasing to 2 weeks, and actually compared to the control if we actually block the regeneration.
Okay. And
do you have, sorry, François, just to link with your question, do you have a way to follow the inhibition and to follow the efficiency of the inhibition, to follow the beta-catenin, for example?
You mean following the location of the beta-catenin? That's what. Right now, we did not test that yet, but one way would be to use the transgenic line, which has the endogenous beta-catenin marked with the M-cherry protein that fluoresces in red when we observe at the microscope.
Explain a little bit better what's going on. It's red and then?
It's red, so hold on, I'm gonna put some slide. Okay, so here are the tests that we made in the lab. Here you can see the DAPI staining. Here you can see the endogenous beta-catenin that it's marked with the protein M-cherry. So you can see at the level of the membrane the endogenous beta-catenin. So when we see the translocation of the beta-catenin from the cytosol to the nucleus, we are supposed to see a co-expression of the DAPI staining and the M-cherry staining here. So we are supposed to see a dot that contains both blue and red. But we couldn't manage so far to see the translocation of the beta-catenin into the nucleus with the transgenic line. It works with the embryogenesis though, but for the other specimen, we are still assessing that.
Thank you, Agata. Sorry, François.
Agata, I was thinking, I was trying to, my head compare to what happens with RNA interference, for example, where you can use real-time PCR data to validate that you destroy your transcript of interest. Is there, and my question was with these inhibitors, do you have a reporter of the inhibition being efficient?
I'm not sure I get the question.
So if you use your inhibitor, basically what I'm getting at is how does it work? What is the mechanism of inhibition?
Yeah, sure. I'm gonna put you the previous slide. So basically, endo-IWR stabilize all these complexes. So technically, the beta-catenin it's destroyed over and over when we use this kind of inhibitor. Whereas when we use ICRT14, it blocks here the transcription factor. If it's
okay.
Was that the question?
So could you, for example, use the beta-catenin protein expression as a reporter? I'm thinking, could you see on a Western blot if beta-catenin is completely destroyed?
Yeah, of course, we could do some quantitative analysis of that, but we didn't do that during my internship. But it would be a great way to have a quantitative assessment of the beta-catenin.
Okay.
Okay, thank you. Stefano?
Yeah, I have a few questions. I believe Paula will stop me when I ask too many. Thank you, Agata, for the nice presentation. So I would start with some technical questions that I didn't I may have missed during the reading your report. So your drugs are dissolved in what, in water or in DMS soap? Because I know some of these drugs So
no,
what did you use as a control for negative control?
Our drugs are dissolved in 1/3 seawater because we are incubating living bisected animals. So we are using artificial seawater 1/3 because Nematostella is a destroyed species. And then we put our drug inside. And in the control, it's just 1/3 seawater.
Okay, so there is no DMSO on the...
No.
Okay, so that was just the details. And then one thing that I'm not sure I understood, because you are looking to look at the proliferation and how the proliferation is changed after your treatment. You basically give EDU from the time of the dissection until the first 48 hours, right? Is it correct?
No, actually, after the bisection, we wait 48 hours.
Okay.
Because it should be the peak of cell proliferation after the bisection of the Nematostella. And after that, we perform the EDU pulse and we fix the animals. And then here I show you.
So how long is the pulse of your EDU pulse?
It's 1 hour.
1 hour. Okay. And a related question is exactly in the photos that you are showing me. So you basically did for your statistics, you did count the number of EDU positive cells in treated and untreated, right, animals. But do you know if the total number of cells in the different specimen is comparable? Because here you have an absolute counting. You don't have a relative, for example, EDU positive versus DAPI in a region of interest. You see what I mean?
Yeah. So, no. The only thing I did, I performed manual counting, as you see, and then for the statistical analysis, since I had here, I'm gonna re-put the slide. You can see there are many. A difference in the distribution number of cells. So I perform a correlation test to see whether it was correlated to the size of the animals, and there was no correlation. So our thought is that it's just a biological process, and it's either depend on the batch, so on the single organisms, or also on the, maybe I took another dividing cycle cell, another dividing cycle. So it was a little bit delayed. I don't know if it was clear.
Yeah. Okay. Yeah, and then I mean, you present in your introductions today that there are 11 or 12 wings in Nematostella, or. And here, these photos means there is a Wnt 7b, but then you have Wnt A, Wnt 2, and Wnt 4. Do they have the same pattern of expression, all these Wts? And do you know if they are, so with the drugs, you block the beta-catenin, but do you have any clue of which of the Wts of the frizzle may be involved in the canonical pathways in Nematostella?
No, we did not perform, for example, a genomic study. So we don't know which exactly is the Wnt, but yeah, I think we were focusing mostly on the Wnt present during regeneration. So here it's Wnt 7, because I also know that the Wnt expression changes, like depending on embryogenesis or regeneration. But we did not go further on the type of Wnt that is expressed.
Yeah. Okay. And then there is one thing that I was trying to, I had to do some of homework, because it wasn't clear to me right away the link between, so you're looking at the Wnt and the effect on the MAP kinase and the ERK pathways, right? So and you're also you didn't show it today, I think, but you did look also at the apoptosis, the effect of the apoptosis of your drugs. So can you elaborate a little bit what is the link? Why looking at the MAP kinase ERK and the apoptosis link to them?
So the first with the MAP kinase, a previous, like in the article here cited of Johnson et al. 2021, they figure out that MAP kinase is also involved in early stage of regeneration, so which is also in line with the cicatrization of the animal after bisection. So we wanted also to see the impact of inhibiting the Wnt pathway on the MAP kinase. So to understand more the network and the logic within these all these pathways that are involved during regeneration. And for apoptosis, also we wanted to see whether there was an impact for sure, I didn't present it today just on the annex of my report. But for example, in Hydra, they already stated that the apoptosis induces the Wnt3 secretion that is necessary during regeneration. So apoptosis is a trigger for the Wnt pathway activation in Hydra. So we wanted to see whether it was the same for the regeneration in Nematostella.
Okay. Okay. Do you have time for a last question, Paul? So. Or two. Well, I saw that you quoted at the beginning of your manuscript, and at the end you quoted Richard Goss, right? And which is a pretty very nice quote. And since you quoted, you know that Goss was basically supporting this is just if you talk about it, right? So. Is supported in a non-adaptive role of regeneration. We thought that regeneration is an epiphenomenon of development, and irrespective of the adaptive value. So he thought that regeneration was. He was suggesting that regeneration was ancestral and lost several times. So it was. Did you think about these or?
No, not really. But when I was reading a little bit about the literature and the quotes about regeneration, I was very intrigued, and I was like thinking all the time. But what is the place for Nematostella in this quote? Because like it's virtually immortal as an organism, virtually like not like scientifically, but still it's a very interesting organism. So is it really accurate, this sentence, if we cut off the development, but like in regeneration, as you said?
Well, if I can say my two cents, I think the place of Nematostella or any other organism that regenerates or not is you can find it just with a phylogenesis with a nice robust tree, and then from there you can start and also bring the Goss quote and try to dig if where it fits in that quote.
Yeah,
so
sure.
Okay, I think it leaves space to someone else.
Thank you very much for
Thank
your
you, Agata.
Thank you, Stefan. It was very interesting. Margarita, do you have some questions on this topic?
I don't have any question, just that I really like the presentation, and a really beautiful presentation. I like a lot how you introduce the topic and the illustrations, and I really enjoyed it. Congratulations.
Thank you very much.
Many thanks, Agata. I think that we can close the question. If there is some other questions from the best jury of the day.
No.
Okay, thank you very much, Agata.
Thank you.